All right. Hey guys, how are you? This is part 2 of microbiology. Let me check. It's showing exactly what I wanted to show. Okay. All right, so we're now going to do a part 2 of microbiology. There will be a total of 6 videos. I'll make 3 before the midterm and 3 after the midterm. Okay, I'll post some potentially earlier, but let's see how it goes. Okay, so part 2. Let's talk about viruses. Let's talk about bacteria. Once again, this is only a limited discussion, right? I've already advised you what to do with these videos. They are a complement. They are just something that you need to pay attention to when studying, right? Obviously, they complement our class. And I often will touch upon a few things in them. So just a few things I noticed. I did re-listen to my previous video. Sometimes, you know, when you're teaching, you tend to say things which make 0 sense listening to them the second time, just like, you know, in conversations, you may misspeak. So I said, what did I call it? I called it ribosomal endoplasmic reticulum, RER. No, it's obviously rough endoplasmic reticulum. That was a mistake I caught while listening to it. There was something else. I think that's pretty much it. The second one, I figured out, I was going through your weekly slides. It actually does discuss this fact that mycobacterium cannot be stained with a Gram stain due to mycolic acid present in the wall. And it actually even lists the dye that is normally used, carbylfuchsin. I don't think it's super, you know, in general, as I mentioned in class, I think Gram staining is super important if you love history. Okay, so let's focus on viruses and bacteria. And for mycobacteria, just know that acid-fast stain is not a method of viral detection, but is a method of mycobacterial detection. Mycobacterium tuberculosis being the most important one. There are actually other mycobacteria, like Mycobacterium bovis, that you may be studying in your pathology courses, internal medicine courses later on. Just know that there's Mycobacterium is the name of the gene. Tuberculosis is the name of the species. All right, so what do we need to know about viruses? Obviously, everything. You need to know that viruses are super small, even compared to bacteria. Okay, so, and obviously, this is generally speaking. A pox virus is the largest pox virus. We just spoke about smallpox, right? So it's actually, but let's stick to this material. You need to know that viruses can be seen with the electron microscope. You also need to know that they can also be seen with a kind of staining called immunostaining, in which antibodies that carry a marker bind to that particular virus, and then you can look for the marker. So let's say this bottle is actually something that you can't see, okay? But let's say this bottle has a cap that you can see. So immunostaining works in a way that if I want to see this bottle that I cannot see, I'm looking for this cap. No, actually, it's not a perfect explanation. So let me just explain it as it is. So there are antibodies that have a part, and we'll talk about antibodies in the next video, by the way, okay? So there are antibodies, you know, they're Y-shaped structures and so on. So what do? They bind to things that are antigens. So a virus is an antigen, and some viruses may have very specific antibodies, okay? So we basically stick something to that antiviral antibody that we can see, okay? So now we're no longer looking for the virus because virus is too small to see, right? But we're looking for that object that we can see that is attached to that antibody, okay? So for example, let me make it like totally crazy. Here is a mouse. Here is a bottle that you can't see. Let's say it's really, really small. And let's say this, am I visible? Sorry, I need to confirm before it's too late that you can all see me. Okay, so let's say this is an antibody, this is a virus, and it's invisible, right? Imagine it is. Imagine it's also very small. So if I were to send an antibody to bind to this virus, it would, but I won't see either one of them because antibodies are also very small, right? Okay, so what do? I attach this visible object to an antibody, and then this antibody binds this virus. I still don't see the antibody or the bottle, i.e. the virus, but I see this little flag attached to the antibody, right? This is the principle of immunostaining. One of them would be ELISA, immune-linked immunosorbent assay, okay? Enzyme-linked immunosorbent assay, or immunostaining with fluorescence. So you use a special kind of a microscope and you look for this little flag, okay? And then you can literally see anything. You can see little proteins bound to a membrane and so on, okay? So you kind of walk around it and use antibodies. It's a very, very commonly used way to detect microscopic objects. All right, so as I mentioned earlier, viruses are non-living, and we call them non-living because they can't reproduce on their own, okay? So they don't have a cell machinery where they're like, hey, you know what, I want to double. In order to double, virus has to infect a living object, i.e. a cell. Okay, and viruses can be isolated and cultured in a cell culture, okay? You can infect cells with a particular virus, and you can culture them, let them reproduce essentially, and then just the number increases and you can collect them, okay? So viruses, yes, they can be isolated in a cell culture. Let's talk about the structure of a virus, okay? Just like cells, just like any living being, they do have a DNA, some of them. But here is where the difference is. You don't have bacteria that just have an RNA and no DNA. You do have viruses that have DNA or RNA. Okay, and there are different kinds of DNA. Some are double-stranded DNA. Some are just single-stranded. Some are double-stranded RNAs. Some are single-stranded RNAs. Some are, you might be hearing about it. So it's an RNA that goes this way or that way. It's all a little bit complicated, and it's more of microbiology, more of biochemistry, molecular biology, not very pertinent to this class. So I'll kind of just mention to you, a virus can be either a DNA virus or an RNA virus, period. A virus can be either a DNA virus or an RNA virus. Okay, not both. Okay, that DNA or an RNA in a virus also has its boundary, but here is the difference. Here is the difference. A viral boundary is a protein, and we call it capsid, like a cap, and add SID, capsid. Okay, capsid is the boundary. And when you see those beautiful, crazy shapes, like looks like a bullet, or looks like a, I don't know, octagon, or looks like whatnot, you know, some extraterrestrial object, those are the capsids. Okay, those crazy looking shapes, those are the capsids. But what you do have to know is that those capsids surround the DNA or the RNA. There might be some little extra protein. There might be some more protein inside that capsid. There usually is, right? Obviously, because that protein will help virus reproduce once it gets into the cell, into a living cell. Okay, so each virus has either a DNA or an RNA. It does have a shell called the capsid. Do the viruses have a membrane? Because we all do. We don't have a cell wall, but we have a membrane. Bacteria, some bacteria have a membrane only. Most bacteria do have that cell wall. That's why they have a very specific structure. What about viruses? Some viruses do have a cell membrane, and we call it an envelope or envelope, whatever you like to say. Okay, I like to say envelope. Okay, so some viruses do have that envelope. Okay, some viruses don't. And you know what's really interesting, and this is a little bit beyond what you need to know, but a really interesting thing is that viruses that have their envelope, i.e. your cell membrane, are actually more sensitive to their environment and not as hardy. Okay, why? Because that membrane can be damaged. Okay, capsid is much hardier. That's why viruses are so difficult to kill in nature. Okay, do viruses have any other organelles? Do bacteria have any organelles, membrane-covered organelles? No. Well, for viruses, it would be hell no, right? Because obviously viruses are even smaller, right? So DNA or RNA, capsid surrounding it with some extra protein. Yes envelope or no envelope, depends. And no organelles. That's it. That's the structure of the virus. The protein covering is called capsid, and it's protective, right? So a few terms that you will definitely see in your clinical life. One term is called viral load. Viral load, L-O-A-D, as in load. Is it heavy or is it light? That's exactly what it means. If the viral load is very high, it means that the amount of virus circulating in the bloodstream of that patient is high. If the viral load is low, that's what we want to be. Remember, blood is supposed to be sterile, right? So then the amount of virus circulating in that bloodstream is obviously low, right? Where is this regularly used? Well, how about HIV, human immune deficiency virus that causes AIDS? HIV stands for human immune deficiency virus, and AIDS stands for acquired, that means you get it, immunodeficiency syndrome. So having AIDS does not, having HIV doesn't immediately make you an AIDS patient. HIV patients that have very, very low T cells are said to have AIDS, okay, the disease itself. And you do need to know how low for your clinical life. And how do you know those things? Well, you know what the normal is. So normal for T cells is between 500 and 1,200. And when the T cell count, the T as in time, T cell count goes below 200, we say the patient has acquired immune deficiency syndrome, AIDS. And that's the patient with HIV. Okay, let's talk about one more virus that probably all of you know, and that virus is called herpes virus. What does it cause? Well, it causes cold sores, right? Here, or as we'll see in the lectures, also on the genitals, because herpes, right? Cold sores here or on the genitals, both. There is herpes 1, there is herpes 2. Herpes 1 can be here or on the genitals. Herpes 2 can be here and on the genitals. That's it, okay? I don't need to explain this. Excuse me. So, cold sores are caused by the herpes simplex virus, Staphylococcus aureus versus Staphylococcus epidermidis. Staphylococcus is a genus, right? So herpes is a genus. So, okay, so herpes is a group of viruses. Herpes viridae, we say, okay? All right, and so herpes simplex is just one virus that causes cold sores, but there are a few other herpes viruses. Okay, and they're all DNA viruses, but maybe I should back up from that. Okay, another virus that you do need to know, and we talked about in class, what is a varicella zoster virus, and everyone knows it as chickenpox. Okay, varicella zoster virus causes chickenpox, and that's not a problem. You see, chickenpox, little kids have them, and that's not a problem at all because they get better. The problem is that varicella zoster virus likes to sleep, and it stays in the body, in the dorsal root ganglion, which is a part of the ganglion. Do you remember what ganglia are? Ganglia is that area where the cell body of a neuron is just outside of the spinal cord. Okay, so dorsal root ganglia, right? Where the nerves basically come back to the spinal cord, right? The ganglia. Now, what's the problem? Well, the problem is that they like to sleep, and they sleep for years. Who likes to sleep? The viruses. The viruses. They like to sleep. Why am I saying it so many times? Because latent means likes to sleep. Comes back late after sleep. Let me clarify. So this virus stays latent, dormant, sleeping, okay, in a particular area of the dorsal root ganglia. Let's say it stays at the level of L1, okay, abdominal level L1, okay. Let's say the patient in whom this virus stayed asleep for, I don't know, 50 years, so this is a little kid who had varicella zoster infection, gets better in no time, okay? After 50 years, this patient has some problem, let's say HIV, okay? Now, his immune system is weakened. Varicella zoster virus that lived in that dorsal root ganglion suddenly says, time to raise my ugly head, because this is exactly what it's going to do. It's going to awaken from its dormancy and cause pain, rash, but that pain and rash is very localized, and that's what zoster means. Zoster is a belt, okay? So varicella zoster virus causes a red belt like this, or somewhere on the face, but it's always in the distribution of that particular nerve, whatever nerve was affected. So If the nerve L1 is affected, the rash would be in the area of L1. If nerve L2 is affected, rash would be in the area of L2, and so on. Okay, so the varicella zoster virus becomes latent and is reactivated into a zoster. What do we call that zoster? We call it shingles. Okay, so varicella zoster virus, little kids get the rash all on their body. Then the rash disappears. Kids are okay, become adults. If immune weakened, have a localized rash. Only one area is affected. What area is that? That presents as shingles. That area where the virus was sleeping in that root ganglion. Okay, so, and just remember that latency is a term that staying low, kind of after, so like asleep. Okay, so the viruses can resurface years after the initial infection. Okay. All right. Now let's talk about a virus that you may have heard about too. All of you have heard about a disease called kissing disease. Okay, it's very popular. It's a catchy name. Kissing disease, another name is mono. Okay, now, obviously you don't get a kissing disease if you're a mono. Just making sure. Okay, you get kissing disease if you're a duo. Okay, so mononucleosis is the complete name. Okay, just for your memory. Kissing disease, another name, mononucleosis. Now, what virus causes kissing disease? It's very easy to remember. How do you get a kissing disease? Well, you go to a bar, you sit there, just someone, and you get the kissing disease. That's it. So it's very easy to remember the name of the virus because the name of the virus is Bar virus. Well, actually, the real name is Epstein-Barr virus. Once again, Epstein-Barr virus causes infectious mononucleosis, another name, kissing disease. Okay, just remember that. I try to make it a little bit funny. It's easier to make those jokes in class, right? Because we're interacting. Here I am. I'm trying to make sure I talk about everything, trying to remember, kind of simplify things. But it's always, once again, the best way to learn is interaction, right? All right. You also need to know about hepatitis viruses, okay? Just know there is virus type A, type B, type C, type D, and type E. That's it. A, B, C, D, E. We'll do more of it probably toward in other videos, okay, if necessary, okay? And the last virus that you need to know, you probably never heard that name before. It's called coronavirus. Coronavirus, I know it's a very obscure name. You probably have never heard about it, but believe it or not, coronavirus has been around for quite a while. It's that variant that we call coronavirus type, Corona 19. That's relatively recent. That we all love so dearly, right? But coronavirus has been a long known cause of the common cold. Just remember, okay? So let me review again. We talked about HIV, we talked about herpes simplex, we talked about varicella and varicella zoster, which is nothing but shingles. We talked about the Epstein-Barr virus. That's what you get when you bar too much, okay? And we talked about the names of the hepatitis viruses, A, B, C, D, E, and the coronavirus that causes common cold. That's all you need to really know to start off your virus education. Okay, and we'll do the rest in class. But let's talk about bacteria right now. Let's talk about bacteria. Okay, this is very important. Okay, so as I mentioned to you in class, bacterial shape obviously depends on the shape of the cell wall behind me. Cell membrane is here. Cell wall is behind me. Cell wall has a shape. My skin is just a wrap. Okay. What did I tell you today is the protein component, the main part, the main building block of that cell wall. The word is peptidoglycan. You need to know it cold. Peptide, a small protein, peptidoglycan. Glycan stands for sugar. So peptidoglycan is the major component of the bacterial cell walls. Okay. So membrane, peptidoglycan layer, which is a cell wall. In some bacteria, not in all, but in some, there is one more layer on top of it, a protective capsule. And that capsule is full of sugary parts, which we call glycocalyx. And some bacteria have a lot of it. And when we culture those bacteria, they actually look very slimy because of that capsule. There is a bacterium called Klebsiella, which causes multiple problems. Like you'll see some of the very, very ill patients on the wards will be coughing up that, mucus with blood, alcoholics, and they will have, if they have Klebsiella, that mucus will be very viscous. And if you culture that bacteria in the lab, you will see, hey, there's so much mucus there. That mucus is the capsule, okay? Like a gelatin-like substance, okay? So I went over this in class, and you should know it by now, week 2, that cocci or cocci, depending on, some like it hot, some like it cold. Cocci or cocci are tennis ball shaped bacteria. It's not that large, right? So if they are in chains, we call them streptococci. So chain of spherical bacteria are called streptococci. If they look like a bunch of grapes, we call them staphylococci, okay? One streptococcus that you do need to know is called Streptococcus pyogenes. Streptococcus pyo always means pus. So pus generating, pyogenes. Streptococci that causes formation of pus. Well, all bacteria do. I guess this guy does it more so. Streptococcus pyogenes it's just another name for group A streptococcus, okay? And that group always stands for a particular way to group bacteria, and it's called Lansfield, just because the person who described it, last name was Lansfield, okay? So it belongs to Lansfield group A classification, okay? But its claim to fame with a name like strep pyo starts with a P, is pharyngitis. Strep pyogenes, group A streptococcus, strep throat. Strep pyogenes, pyo, P, like pharynx. Strep pyogenes, group A streptococcus causes pharyngitis. Another name for pharyngitis, strep throat, nasopharynx, oropharynx, laryngopharynx. Now, what is a rod-like bacterium called? Rod-shaped bacterium. It's called the bacillus. Bacillus is actually a name of a particular genus. So like Staphylococcus is a bunch of grapes, Bacillus is actually a genus of rod-like bacteria, and the famous species is Bacillus anthracis. Anthrax is a kind of a bacterium that is normally associated with the so-called wool sorters disease. Okay, so it's not really dangerous, and it causes like you know, pustules, somewhat like little areas of dead skin we call on you know, extremities that we're exposed to. That's not the problem. The problem is when you inhale it. Anthrax can be deadly. Okay, very, very dangerous. Anthrax. And you know, laboratories have cultured some weaponized grade anthrax, and that's what some people are mortally scared of in terms of bio warfare. Okay, so anthrax, Bacillus anthracis is rod shaped, and if they are in chains, we call them rod-shaped bacilli, rod-shaped bacteria that are known as bacilli that are in chains. And the word for in chains is what? Staphylococci? Of course not. Staphylococci means bunch of grapes. Streptococci. So streptobacilli. Streptococci are tennis ball-shaped bacteria in chains, and streptobacilli are rod-shaped bacteria in chains. Okay, so you do need to know how bacteria divide. There are few ways. One of them is called binary fission. Okay, so binary fission is a form of reproduction for bacteria. It's an asexual, no sex between bacteria, which is conjugation, is involved. We're not discussing that, okay? Asexual reproduction, and it's very similar to mitosis in eukaryotes, right? Our cells, sometimes what happens, just, you know, cells form, just break down the middle and separate. That's mitosis. Binary fission is the same thing. Bacterium just divides in the middle and that's it. And so the cell divides into half for each. And what does it share among these two cells? It's DNA and whatever organelles it has, ribosomes, and so on. So because DNA has been divided equally, these two cells that result are genetically identical, no difference whatsoever, and therefore no genetic variety is introduced, okay? That's how bacteria can divide by binary fission. Okay, so what if bacteria cannot reproduce very well? What if the environment is such that bacteria think, hey, you know what, if I start reproducing now, not only am I going to die, but my kids that I generate are also going to die. Well, in that case, bacteria do something really interesting. Not all bacteria. There are bacteria that can make this structure, and I'll say what it is in a second. Okay, so what the bacterium does is it packs its bags. It takes all of those things that you can't really live without, which is what? DNA, more of DNA, everything DNA. Okay, what else? All the proteins without which it can't survive, all the things and everything that a bacterium needs to survive. Everything that is not so important gets pushed to the side, and between the part that is absolutely essential to survive and those things that are not very vitally important, the bacterium builds a double cell wall. We call it a spore, S-P-O-R-E. So some bacteria can produce these spores. They contain very, very hardy substances and become extremely hard to kill. Okay, and these bacterial spores can survive very, very, very difficult environmental conditions. no water for years, etc. Okay, and basically, when you think of a spore, think of a dormant state, sleeping. In viruses, we have that latency, right? So it takes time from the time varicella infection happens to the time that the zoster appears, the shingles appear, right? Here we have a time between the spore formation and when water, food finally appear on horizon and actually are available to this bacterial spore again. Okay, that's the dormancy. Okay, so in between, during this time, it's very hard to kill that spore. Okay, how do you? They're the hardest microbial structures to kill. How do you kill them? You kill them by using the autoclave. Some of you have seen it, some of you have not seen it. Okay. What autoclave does, it raises the temperature beyond boiling and adds a lot of humidity. Okay, so that's the way to kill spores. Cells die way before that. Spores, no. Can you boil spores? Sure you can. Are you going to kill them? No. Okay, autoclaving is much harder on the spores and kills them. Okay, so alcohol, if you use it on skin, is not going to do anything to the spores. It's very effective in killing bacteria, does nothing to the spores. Okay, so just because I'm talking about these hard to kill products of bacteria, the spores, I want to mention something else. I've been talking to you today about how we have DNA, how we make RNA from DNA, and how we make protein. DNA, RNA, protein, DNA, RNA, protein. That's the dogma of molecular medicine, of science, essentially. This is how we know life as of today. There is one exception. There is one exception that's called, and that's something to do with infectious proteins. And the word for that is a prion. Prion actually stands for, PR stands for proteinaceous, I stands for infectious. Okay, so prions are proteinaceous, infectious particles. Proteinaceous, what does that sound like? Like protein, and it is protein. So prions are abnormal proteins, usually in the nervous tissue, usually in the brain. Okay, how do you treat against prions? Well, you burn down everything, incinerate. You can't. There's no treatment. Even autoclave is worthless. Okay, so how do patients get prion diseases? There are very few. Creutzfeldt-Jakob disease, CJD, written as, you got to see the name because it's very difficult to relate to if you're not used to it. Okay, Creutzfeldt-Jakob, Jakob is written as Jacob, okay, with a K. So Creutzfeldt, you literally like need to see the name. Okay, so Creutzfeldt-Jakob disease, CJD. There's also another disease called Kuru. But what you do, and then there are a couple of, but what you do need to know is the Creutzfeldt-Jakob disease. Okay, it's the most popular one because of what? Because of mad cows. So about 30 years ago, I think, there was a another panic created by our hardworking journalists, and they talked about mad cows, and they said that in about 10 years, and they showed those beautiful graphs, millions of us are going to die because of that couple of those couple of cows that had mad cow disease because we ate them, okay? And obviously, they slaughtered a few cows, a few hundred cows, if not more. Nothing happened. Like there were a few cases, very, very rare, of Creutzfeldt-Jakob disease, and the story died. Okay, you may see many more stories in the past that have been overblown like that, and this is something that's going to happen in the future as well. So just relax, okay? Creutzfeldt-Jakob is very rare. How do the patients? So yeah, I'll talk about it a little bit more in my my other class. So that's it. How do you get Creutzfeldt-Jakob? Well, by eating brains, obviously, if you do. Not too many of us do that, but you know, because that infectious protein can be transmitted via corneal transplant or dura mater. Remember, today in the class we're talking about the meninges, dura mater, the top layer of the meninges. Sometimes you need to graft those, right? And dura mater grafts could do that potentially, but also contaminated growth hormone, because it's part of the brain, right? Contaminated, not so much recently anymore though, because growth hormone is obviously recombinant. So it's a very, very old information, but you do need to know it sometimes, right? You know, it might be asked of you, right? So obviously equipment, so neurosurgical equipment, right? So once again, it's very, very rare, okay? All right, so, but once again, DNA, RNA, protein is not fulfilled here. Here we have a protein that does what? So let's say I'm an infectious protein. This is a healthy protein right next to me. What do? I do, hey, this protein now becomes infectious. This healthy protein now becomes infectious. What does it do to its neighbor? It also does, hey, nudges the protein right next to it, and that dude also gets to be infectious. Okay, so that's how you get Creutzfeldt-Jakob disease. Its claim to fame. It's a very bad disease, by the way, if you get it. Okay, it's a very, very terrible disease. There's a startle myoclonus. Obviously, I mean, the brain looks like it's been shot through with a shotgun. You have that like vacuoles. You have those little bubbly spaces throughout the whole brain. It's very don't forget, it's a very rare disease. Okay, Creutzfeldt-Jakob. Just yeah. So I mentioned this in class. I really do want you to know some terms. Okay, so just like I was talking about, you know, basic terms earlier on in the prior video, I want you to know, like, even things like anaerobic. What does anaerobic means? No air. Try to break down all those terms. Develop a habit of not blindly memorizing, because memorizing might become very difficult if there is way too much to memorize. So organizing things will actually help you. Okay, anaerobic, anemia. What does anaerobic mean? Aerobic means containing, you know, air, and anaerobic means no air. Anemia, what does it mean? No hemia, no blood, right? And so on. Just be very familiar. So bacteria that, for example, don't need oxygen to survive, they're called anaerobic. You need to know those things, right? Now let's talk about a very important thing that will prepare you for the next week. Okay, let's talk about, I kind of mentioned it, but let's, in class, but let's revisit it in depth. There is a term called sensitive. There is a term called resistant. Resistant, we use the word resistant when there is no antibiotic response. What does it mean? I give you an antibiotic. And when we talk about an antibiotic, what does an antibiotic mean? Because that's if you think like that, stepwise, it's very easy to understand it. You just resist. Resist means what? You don't fall for the problem. You see, the problem is it about you or is it about the bacteria? Okay. So an antibiotic means anti-life. Okay. Anti-life. Anti whose life? Yours. I'm going to give you an antibiotic to kill bacteria, obviously, right? So when I give a patient antibiotic, I obviously want to kill the bacteria. Antibiotic that is supposed to kill bacteria. Fine. If a bacterium resists the work of the antibiotic, it's resistant, period. That's simple. Okay. If a bacterium responds to that antibiotic activity, i.e. dies or stops multiplying, that's called sensitive. It's sensitive to the medication. Okay. I'm sensitive to noise. What does it mean? I don't like noise. It irritates me. Okay. I'm resistant to, those cartoons always say, I'm resistant to your charms. That means your charms aren't working. Okay. So, all right. Resistant. So if you have an agar plate, right? And if I put a medication on it, let's say I take a bleach pill, literally bleach. We all know that bleach kills bacteria, right? So if I take a bleach pill, put it smack in the middle of agar plate, and then plant bacteria. So plant the bacteria, right? And put that bleach pill smack in the middle of the agar plate. We know those pills are deadly to every single one of those bacteria. So are bacteria going to grow right around that pill? Climb on top? No, of course not. They're going to stay as far from it as possible. As far as that little pill can reach its tentacles, diffuse, right? So resistant means growth around the pill is absent, no growth. Okay, resistant means no. Sorry, I misspoke. Let me correct it. Okay, it's kind of getting late. We had a 2-hour class already, so I hope you forgive. Okay, let me bring it back. Resistant means I don't give a damn, right? I can resist. So if you put, forget about that bleach pill, okay? Forget about that bleach pill. If you put an antibiotic, okay, and I'm a bacterium and I resist the antibiotic easily, what's going to happen? That antibiotic has no effect on me, right? So a bacterium, if I were a bacterium, we're going to grow. How far are we going to grow around that pill? Well, we don't care about that pill. We think that pill is harmless, so we can resist its activity. So depending on how much we don't care about that pill, we can grow right up to the pill. For example, I put a pill of penicillin around bacteria in a solution of bacteria that has beta-lactamase. Penicillin only works on bacteria because it has that beta-lactam ring. It prevents formation of the peptidoglycan, right? And so cell wall is not made. What if you do have that beta-lactam enzyme, beta-lactamase, and you don't care about penicillin activity, right? So what happens? Bacteria will literally start growing around the penicillin pill on top. So here's an agar plate, smack in the middle of it. Assume there is, and I'll show you pictures in class, okay, in the next lecture. Let's say there is a pill, penicillin pill. What's going to happen? You're going to have growth everywhere. everywhere, no problem, right? So growth around the pill, even on top of the pill. With fungi, no problem. They don't give a damn about, they'll literally grow everywhere, okay? So that's if the bacteria are resistant, and so there is no antibiotic. So they have a response that where they say, hey, you know what, I don't care about you. There is basically zero antibiotic activity, okay? So if a bacterium is resistant, another word you can say, not sensitive, okay? It means it can grow normally. If resistant, it means not sensitive. If sensitive, it means not resistant. I'm sensitive to noise. I'm not resistant to noise. I don't like noise. Okay, clear. Okay, so let's say I put a disc of bleach. Now let's go back to where I made that little mistake. Okay, I take a pill of bleach. Forget about antibiotics. Bleach kills everything. Okay, bleach kills everything. So you put a pill of bleach smack in the middle of that colony, right? What happens? There is a ring that forms around the bleach pill on agar. Everything is destroyed, dead. The rest of the agar plate is still growing with the same bacteria that you planted, right? Just around that bleach, but a good wide circle around that bleach, you have zero growth, okay? So just remember, if resistant means not sensitive, okay? If not sensitive, it means resistant. But if sensitive, it means not resistant, okay? All right, so just remember this area. So, and I'll show you the pictures, and you have pictures available to you. Obviously, this is a review. I don't want to be showing pictures. It's gonna, you know, become too long, too lengthy. This is a focus review, okay? So just know that when I add that disc, that of the pill in that agar, on that agar plate, that chemical starts diffusing out, right? You know, the little pill, some of those chemicals just move further away down the concentration gradient, right? So if there is nothing, if there is a large zone of nothing growing, we call it a large zone of inhibition. It inhibits the growth, okay? And we say bacterium is sensitive to that antibiotic. Another word is bacterium is susceptible to treatment by that antibiotic, whatever you use it, okay? So if I use penicillin pill and I'm trying to grow, you know, for example, well, there are very few bugs that are actually responsive to penicillin pure. Let's say the one that's pretty much responsive is syphilis, okay? So if I see a lot of syphilis bacteria around it, that means those syphilis bacteria are not sensitive. They're actually resistant because if they're growing, right? But if I see that syphilis bacteria are like, ooh, no, I don't want to come close to that antibiotic. It's going to kill me. It's going to harm me. What is that called? It's called sensitivity. Okay, it's sensitive to that disturbance by the pill. Okay, so you need to just keep on explaining those things to yourself. It's easy to stumble because it's not something we use every day. And don't think about I'm resistant. Think about bacteria, right? If bacteria are resistant, that means there is no zone of inhibition. They can literally climb on that pill. They're resistant. They don't give a damn about it, about any particular antibiotic used, right? So once again, the best way to remember is if you use bleach, chlorine disc, right? If you place it, it will make a very large zone of growth inhibition, right? And that's the way to remember. So bleach obviously is not an antibiotic. It's a sterilizer essentially, right? It's going to kill everything around it. Can you say bacteria are resistant? No, hell no. They're very sensitive to bleach. Respect, big time, right? No growth. Because why? Because you're going to die otherwise, right? Okay. So just read it a couple of times and you'll get it. Okay. All right. Another way to so bleach obviously you don't develop too much of a resistance to that. It's a chemical process that's very destructive to bacteria. Okay, luckily because a lot of bathrooms are clean because of that bleach, right? But hey, how do bacteria increase their ability to be resistant to bacteria? Because antibiotic resistance is very important for bacteria, okay? Bacteria absolutely need to be resistant. Why? Because bacteria do want to dominate us. It's either us or them. And it's not only about us. You see, it's the whole survival thing, right? So bacteria want to have as much antibiotic resistance as possible. Now, how do they do it? Well, there are multiple ways. One of the main ones is the gene mutations. Okay, so mutations, sometimes multiple mutations in all of those microbes are very important. Okay, but you'll hear a lot about it in your week 6, 7, and 8. Okay, it's the term drug overuse or misuse. Okay, and it's also very important. But you know, it's a double-edged sword. It's very easy for those that don't work at the hospital say, do this or do that. In the clinical world, it's very difficult to not prescribe antibiotic, you know, because and I'll probably go over it in class a little bit. Okay, so I don't want to go into detail. Just remember, one way is how bacteria acquire this antibiotic resistance is just via gene mutation. So bacterial gene mutation, DNA or plasmid mutation. Another name is via overuse of drugs or misuse. And I mentioned today in class that urinary tract infections were in females were originally treated with sulfa drugs, right? Sulfamethoxazole was the medication, TMP-SMX, trimethoprim sulfamethoxazole. Then after a while, it just stopped working. So what was used after that was a very good antibiotic called ciprofloxacin. And you've all heard the word cipro, right? Ciprofloxacin. Well, guess what? Right now, it doesn't work at all. So we use amoxicillin. And eventually, amoxicillin will stop being effective too. That's why it's very important to have those backup medications. Okay, but just remember, antibiotic resistance is unavoidable. Eventually, all microbes, bacteria will catch up and become resistant. That's why new generations of medications are important. That's why we need to return to and identify other ways of treatment. Believe it or not, initially, before bacteria were treated with antibiotics, there was a lot of research done on how to treat diseases by using viruses, actually. Okay, so viral cultures, etc. Only that method was very lengthy and slow, and some of them worked pretty well. Okay, some of that work is kind of almost esoteric right now, but people are working into bringing it back. Okay, because more and more medications are becoming worthless. So at some point, you want to find a way to, treat in a different way. Okay, but as soon as antibiotics came on the market, penicillin immediately saved so many lives that we essentially stopped thinking about that lengthy treatment that didn't always give us clear-cut results, right? So I told you, antibiotics and vaccines are the reason why we enjoy our quality of life, long life, and life essentially free of nagging little diseases. Okay, pneumonia became much less of a problem. Antibiotics, and so on. Okay, so okay, so when, just because you need to know it, right? When you're discharging a patient home, always remind to complete the course of antibiotics, because those patients that are released from the hospital and just stop taking their medication. Okay, I'm already better. I've taken one pill. You're supposed to take like 20 pills, right? That's not going to work, because that first pill will now become essentially a way to train those bacteria to not care about that antibiotic anymore. For example, I give 1 pill of Cipro, but I have to treat with 10 pills of Cipro. But after 1 or 2 pills, a patient already feels much better. So the patient thinks, hey, you know what? Maybe a bad idea to take 10 or 20 pills of Cipro. Maybe I'm harming myself. That's the notion among many of us, right? Me included. I mean, I used to think that way too. Okay, so always finish your medication, right? Always finish your medication. All of the prescribed course of antibiotics should be finished. That reduces the risk of antibiotic resistance, disease recurrence, and so on. Okay, so don't forget a very important regulation. When you start working in the hospital, you will always have a patient coming to you with a high fever, for example, right? High fever, and you know the patient has this or that disease. Let's say you can diagnose them. So what are you going to do? Antibiotics, right? No. Before you treat the patient with antibiotics, okay, you need to collect the specimen, whether it's the blood or sputum or urine or anything, okay? Obviously, for that, common sense, use sterile supplies, collect the specimen in appropriate transporters, in appropriate containers. But please make sure you collect those specimen before the antibiotics are started. Okay, but what happens if, like, you have a patient and that patient comes, you truly care about your patients, let's say you don't know what the diagnosis is, okay? But, the patient is coughing. I showed you the slide today, pneumonia. So what do? You use the broadest spectrum antibiotic that you can think of. Amoxicillin, for example, is a good broad spectrum antibiotic. What does broad spectrum mean? Kills many bacteria, most and all bacteria that we can suspect of causing that particular disease. That's the broad spectrum antibiotic. It's not just kills one particular bug. For example, your penicillin. Penicillin is used only for syphilis, essentially in 2022, strep throat too, but we don't want to use it for strep throat. Why? It doesn't work very well. There's a risk. Where it does work foolproof, and in fact is the only way to treat the disease of syphilis. Okay, so how do you treat a patient with syphilis? With penicillin, But what if someone comes to you and you don't know? It might be syphilis, it may be something else, then broad spectrum, period. Okay, so penicillin, well, syphilis is a bad example because it's relatively easy to diagnose, right? But, and I'll show you how. We'll actually do it next week, okay? So broad spectrum antibiotic, if you don't know the specific diagnosis, which, but, well, you would know the diagnosis, you just don't know which bug it is, right? So you give the medication that covers all bugs that cause that particular disease, okay? And just know that most antibiotics, so first of all, if you don't know what the pathogen is, which bacterium it is, use the broad spectrum antibiotic, but also know that most antibiotics do not kill bacteria at low concentration. Most are so-called bacteriostatic, stop, stop, okay? Bacteriostatic. Most antibiotics are bacteriostatic at low concentrations. If you take and inject the whole pill into blood, and you're only supposed to give like this much, you're gonna kill all the bacteria. Your patient will also die, but you will definitely kill all the bacteria. So you don't want to do that. Your patient might get harmed. Remember the story of methicillin. It's very good. It killed Staphylococcus. It also killed the patient. We want to be very cognizant of the side effects, right? We don't want to harm the patient. We want to help the patients. Okay, so most antibiotics are bacteriostatic at low concentrations. Bacteriostatic means it stops the replication of, or stops or inhibits, okay, a replication of bacteria. So there is no division, okay, no new growth. And then the body takes care of whatever is there, okay? Now, what's the difference between bacteriostatic? What's the other one? Bactericidal. How do you remember it? Suicide, boom. Suicide. Suicide means you kill yourself. Side. So bactericidal, you kill bacteria, period. Antibiotics, which kill bacteria, are called bactericidal. Easy, like suicide. Okay? Okay, so very important for your pharmacology class, very important, is a term called MIC, which stands for minimum concentration. Minimum concentration. I stands for inhibitory. Means it stops. So concentration, the minimum concentration that inhibits the growth. MIC, concentration, that is the minimum, the lowest concentration that still works. Minimum inhibitory concentration. So when you have that solution, we call it the broth, right? And you dilute your medication, the tube that has the lowest concentration of medication, okay, will give that to you. So you measure the concentration of the medication that is the lowest, but still does the job that you want, okay? That's the minimum inhibitory concentration. Very important for your farm. Please remember it, okay? Minimum inhibitory concentration, MIC. All right, and the last point for bacteria. Remember, you're going to be using those agar plates. There are test tubes with agar put at the bottom of the test tube. It's like a slant agar, test tube slanted agar, and both of those are the same. And agar is considered to be a solid medium, and you know they want you to know everything about how to culture bacteria, and you're going to know it. We're going to do it in the class, right? So when inoculating a sample agar culture, just go through your rules and know them. keep your lid one hand during inoculation, flame that loop. Obviously, you never flame the loop these days. Why? Because you get fresh single-use plastic loops, right? But if you do use those metal tubes, and if you do use those Bunsen burners, what do? You ensure that it glows red, okay? And then don't play around with it, don't stab the auger, and so spread sample in a specific pattern. You need to know that you got to divide those plates into 4 quadrants, and then you kind of like separate from you kind of try to avoid mixed cultures, and you do that by separating them into subcultures until only one species is present. How are you going to see that? Well, you can actually recognize species, they have individual growth, right? So, and you know that the plate is stored with that auger on top. So not with the lid on top, but with the auger on top to prevent dripping into colonies, because bacteria would just start like swimming in there otherwise, okay? So auger side up in the incubator for optimal growth. Some people think it's important that you know these. And this is a part of your course, so know them. Know everything I said just now. I'll try to pass out, pass out the notes to help you kind of deal with it. All right, the last part of this review would be talking about parasites. Okay, parasites are one cell. What's another word? Unicellular, one cell, or multicellular, many cellular, many cells, unicellular or multicellular organisms. How do you study parasites? Well, there is a science that studies parasites. It's called alogy of parasites, parasitology, mycology, study of fungi, parasitology, study of parasites. Okay, so there is a term ecto-endo. You know, ecto means outside, right? So ectoparasites are, for example, important for transmission of parasites, which cause diseases like Lyme disease, malaria, and Rocky Mountain spotted fever is something that you should have heard about, abbreviated as RMSF, Rocky Mountain spotted fever. It's actually not found in the Rockies. It's on the east coast of the US in North Carolina area, okay? So it's just a name, okay? Rocky Mountain spotted fever is transmitted via a tick called Lone Star tick. Lone Star tick. I think it's called Amblyoma americanum. You can Google it. Lone Star tick. Okay, Rocky Mountain spotted fever. If a vector, if something that transmits a disease is a living being, we call it a biological vector. Okay, so this tick, the Lone Star tick, call it a biological vector. Okay, so what about another ectoparasite vector, another biological vector? Well, another one would be a mosquito. Mosquitoes, just like the Lone Star tick, are ectoparasite vectors or biological vectors. But when mosquitoes drink a blood sample that contains parasites called plasmodia, okay, plasmodium singular, they get some of those plasmodia, and when they drink blood again, those plasmodia are passed on. Okay, they obviously have a cycle within the mosquito. So Plasmodium then infects the red blood cell, and the cycle of malaria starts. Okay, Plasmodium is a so-called protist. Protist means a pathogen. Protist pathogen. So does it have a nucleus? Yes, all protists do have a nucleus. It's a eukaryote. Okay, it's eukaryote. And what does it mean? It's a protist. It means it's not an animal. It's not a plant. It's not a fungus. It's a protist. Okay, so and it does cause malaria. Okay, so Plasmodium. All you need to know is Plasmodium protist that causes malaria. Who transmits Plasmodia? The mosquitoes. So mosquitoes give you Plasmodia. Okay, and Plasmodia is a protist that causes malaria. Is it a plant? No. Is it an animal? No. Okay, is it a fungus? Also no. It's a protist. Okay. What about Rocky Mountain spotted fever? So ectoparasite called Lone Star tick is your vector, the biological vector, right? And it transmits the disease. Okay. So all right. So bite of them, just remember, okay, bite of an infected mosquito represents, disease transmission via a biological vector. All right, I kind of mentioned it today in class multiple times. Metronidazole, you sure do remember metronidazole. And I told you need to know it, and you do. Metronidazole damages the DNA and kills the target. Okay, so when is metronidazole used? Well, I told you that all infections of the abdomen, anaerobic infections of the abdomen and vagina, many of them can be treated with metronidazole. This is just a generic fact. What about the term that I mentioned called Giardia or Giardia? Giardia. What is a Giardia? Giardia is a parasite most commonly infecting human gastrointestinal tract. Giardia is a protozoan. It's like the first, proto means first, zoa, living being. So it's like a little animal, the first one-celled animal, right? Giardia. How do you get it? You get it by eating contaminated food with Giardia or contaminated water, okay? And I'll give you a few other ways they're gonna ask you, okay? So Giardia, full name is Giardia lamblia. They might also talk about a different bug called Entamoeba histolytica. Entamoeba histolytica, but Giardia is more commonly asked. Okay, so a person who has Giardia has severe diarrhea. What happens in diarrhea? Let's think about it. Severe diarrhea. So you go to the bathroom multiple times. Do you lose stool? Yes. Do you lose what is the definition of diarrhea? And it may sound funny for most, but you do have to define things before you can treat them. Okay, so when the stool takes the shape of the container, and passage that is very common, stool takes the shape of the container. So when does stool take shape of the container? When it's very watery. So if it's very watery, are you losing water? Yeah, when you're going to be dehydrated. So let me ask you, how are you going to treat a patient who is dehydrated? Are you going to give him fluid, or are you going to tell him, hey, sunny outside, we're in high heat summer, let's take him to Vegas and ask him for a walk, right? Obviously not. The patient is dehydrated. So you give that patient fluids and electrolytes. But did we say Giardia? Yes, we did. We treat Giardia or Giardia with metronidazole. Giardia, metronidazole. Giardia, metronidazole, and I'll tell you how to remember it in class, okay? Giardia, metronidazole. All right, so you do need to know a term Candida. Candida is a genus, and there are many, many species. One of them is Candida albicans. It causes thrush, and there is a picture I'm going to show you in class. Thrush, it's like a fluffy white covering on the tongue, as if a person is eating like that Greek yogurt or something and just has it on their tongue. So, okay, there are quite a few fungi that you need to know. Okay, so I mentioned today a fungus called Aspergillus. Aspergillus flavus is a fungus. Is it Aspergillus or Aspergillus? Aspergillus, I think. It's getting late, guys. I just don't want to think about it. I want to get it done with. I like to get things done way ahead of time. I feel happier then. Okay, Aspergillus, Aspergillosis, Aspergillus. I do need to, yeah, that's what happens when you're like totally tired. You don't know what to say anymore. Okay, so Aspergillus, yeah, definitely. It's Aspergillosis. Oh my God, I can't believe it after talking about it for years. Okay, so Aspergillus. Okay, so it's a fungal fungus. Okay, it's a fungus that makes mold, right? And you see it on bread, and you do have those allergic reactions when you inhale the spores. Okay, and don't forget. Hepatitis B vaccine strongly recommended. Less risk of liver cancer caused by this mold. Okay. Another one you do need to know is a thing which is a mouthful. Coccidioidomycosis. Coccidioidomycosis, abbreviated as cocci usually. And where is it found? It's actually a lung infection from around the corner in San Joaquin Valley. Okay, used to be known as San Joaquin Valley fever. It's a fungus that is dimorphic, means it has two different shapes. It's present as a mold when it's colder, and as yeast when it gets really hot. Okay, and humid. Okay, so the fungus is called Coccidioides immitis. Okay, so coccidioidomycosis. I know it's kind of hard to remember. Aspergillus and coccidioidomycosis. Hey, I mean, after talking about these for years, I didn't know anymore. Is it Aspergillus or Aspergillus? When you guys are tired, I would not worry about not remembering the names. It comes and it goes. Hey, you use it or lose it. Don't feel bad about it. But here are two things, the last ones before I round it up, this part of the video. Let's talk about thrush. Okay, thrush is caused by Candida. We all know that. Candida and species would be albicans. But and thrush, Candida is a fungus, but here is the deal. There is a thing called ringworm, reddish rings on the skin. It's called ringworm, and it's caused, ringworm disease is caused. So first of all, let me segue into this. Who gets ringworm? Athletes, right, on their foot, athlete's foot. and so on. And despite the name, this is nothing to do with a worm. It's a fungus. And you need to know this. You need to know the name of the fungus. It's called tinea, Tinea. They have, it's called dermatophyte. It's a type of a fungus, okay? And it has that species name to it, like Tinea corporis, Tinea cruris, Tinea whatnot. Corporis means body, okay? So tinea of the body is the fungus that causes body ringworm. So those rings, okay? So once again, ringworm and candida are not the same, but both are fungi. Okay, fungus, plural fungi, or fungi. Some say, I'm sure there are people who say aspergillus as well. I mean, seriously, otherwise, I'm pretty sure, like, even if tired, I would have kind of like, I probably heard it in both variations. And so I don't know anymore what I usually say. I think I usually say aspergillus. Whatever I said in the class when I was not thinking. So, okay, so thrush caused by candida. Fungus, Tinea corporis, causes ringworm. These two are not the same, but both are fungi. Aspergillus and coccidioidomycosis. And good luck trying to learn how to pronounce coccidioidomycosis, because it's an important lung infection that does happen around the corner in San Joaquin Valley. Okay. All right. I think I will round up this this part of the recording right here.